honestly, that word metabolomics, right? It's metabolites, omics. There's a lot going on there, and it is actually an exciting word in many ways. So demystify it for us. What exactly does it even mean?
Yeah, absolutely. And the demystification really, Casper, is so easy. Metabolomics, as you said, metabolites and omics. What is an omic science? It is a complete set science. It is a totality of something. So when we say genomics, we're referring to the complete set of genes. When we say proteomics, we're referring to the complete set of proteins in a body, in a system. Really, it could be anything. Let's say a human body. And when we say metabolomics, we're referring to the complete set of metabolites. And so metabolomics is essentially the study of the dynamic interplay of metabolites in your body or in a biological system in general.
And why is that important to personalized medicine? Because this is one of my pet peeves is everyone likes to say personalized medicine. It's a very key word these days. But when you dive into it, most of the conventional approach to personalized medicine is looking at some kind of marker that is personalized to you and then going with something that is very unpersonalized in the treatment side. And again, that marker may be somewhat limited. So how is this more personalized than what most of it is out there already?
Yeah, so with a lot of current products, genetic testing is been, of course, touted as one level of investigation, and certainly it provides very deep insights that are important. And when combined with something like metabolomics, can add increased insights. But what we see metabolomics as, and it's very interesting to, I was reading the article you wrote yesterday on the death of a diagnosis, and in the end there was this quote by a founding professor of Johns Hopkins that it's far better to know what kind of person has a disease than what disease a person has. And when you look at a person, what is a person? They are the complete set of their systems biology at a molecular level. Now, of course, you can talk about some metaphysical things in there as well, but at a molecular level, as a physical construct, we have to understand a person through the central dogma of biology, which starts at the genome and has RNA that is transcribed from that genome. And some RNA is expressed as proteins, and they're translated into proteins. And those proteins undergo various changes in the body, and the metabolic byproducts of those changes are the metabolites. So when you look at a person as a hierarchy of these systems levels, you see the genome, the transcriptome, the proteome, and down through the metabolome. The metabolome sits at the lowest level. It is also nearest to the phenotype. So it is immediately upstream from the environment and downstream from other levels of systems biology up to the genome. So it is quite literally the metabolome as a level of systems biology. It is at the nexus of those gene environment interactions. And when we talk about the greatest diseases and disorders and the greatest contributors to human mortality and morbidity, we're talking about incredibly heterogeneous disorders that do not have their root purely in environmental factors and do not have their root purely in genetic factors. Most disorders are not genetic disorders. They're genomic disorders. They are omic disorders in the sense that they relate to all systems biologies. Very few disorders have genes with such high penetrance that somebody who has that gene will necessarily develop that disorder. Most disorders, and as I've mentioned, the ones that have the greatest contributors to human morbidity and mortality, those are heterogeneous disorders that are a combination of gene-environment interactions. And again, the metabolome provides an incredibly attractive level of investigation because it sits at that intersection of the gene and the environment.
So let's talk about that investigation because it really is fascinating. And I love the idea of going upstream, whereas most people are looking downstream already and trying to guess the upstream in a sense. And that, again, to me is not personalized or very scientific. It's throwing darts in the dark sort of thing and understanding the end result, but not understanding what the whys or root causes are. So with Therium, what are you actually looking at within the metabolites and the metabolomics to start to get a better picture of the person?
Exactly. Thank you. Yeah, it's a great question. So we, in addition to, 75 different demographic and clinical variables that we collect from our customers via self-report. Our metabolomics test, which we have trademarked the Aristotle, it is a test of sentinel aqueous metabolites, specifically 126 aqueous sentinel metabolites that are found in central cellular physiology. And we have strategically picked out these 126 metabolites to monitor, as they provide for roughly 70% of monitoring of all human metabolism. When we talk about metabolism, at least at the science and research level, we talk about canonical pathways. Pathways such as the TCA cycle or aminoacyl tRNA biosynthesis, phenylalanine metabolism, whatever is canonized. According to different databases, you have between 84 and 99 true pathways that have been studied, typified, and canonized in databases such as the Small Molecule Pathway Database or the Kyoto Encyclopedia of Genes and Genomes, K. And both are great resources. So of those typified pathways that are known to us in current science and technology, we are able to reliably monitor, and by reliably, I mean with at least 2 entries or more, we are able to reliably monitor roughly 70% of all human metabolism. And we do that using a dry blood spot test, which is an at-home collection method. Our customers receive a dry blood spot kit with a finger lancet, and they apply just a few drops of blood to the proprietary filter paper. They place it in our biohazardous aluminum foil pouch with a desiccant, special desiccant inside to maintain metabolite integrity via transit. And they send it to our lab where we process it. And we process it using gas chromatography mass spectrometry. So this is a very advanced analytical technology that, although has many clinical applications, and there are clinical mass spectrometry labs throughout the country, it is still not a mainstay healthcare device. Everything from the footprint of these machines to the specialized knowledge necessary to operate them requires usually a staff scientist, somebody with a PhD, if not multiple people with an PhD, and maybe an engineer to monitor the system when it goes down and maintain it. So they are not currently feasible technologies that are readily deployed in hospitals, and definitely not health clinics. And what we are offering is essentially a research level platform that we apply, coupled to a very simple, at-home, minimally invasive, and cost-effective sample collection method to reliably return concentrations of these 126 sentinel metabolites that are indicative of your central physiological processing. And again, I say 70% of your metabolism can be reliably monitored with our test.